Hello and welcome to Biocapsules. I am Shivali Singh and in this video tutorial we'll discuss about the transposable elements. So let's start. Transposable elements are those DNA sequences that have a unique ability to move from one location to another location within a genome. And this process of mobilization of transposable element is known as transposition. Since these elements possesses a ability to transfer themselves from one place to another within a genome, they are also known as jumping genes, mobile genes, or controlling genes. For the very first time, these elements were identified by Barbara McClintock while working with the corn plant. Next is transposable elements are identified in almost all the living organisms, whether they are prokaryotic or eukaryotic, and they are present in very high proportion in a genome. For example, 50% of the human genome is made from transposable element. And likewise, in maize, the transposable element constitutes around 90% of the genome. Since these elements are present in very high copy number, it is hypothesized that they might have a role in change in genome size during evolution in plants, Drosophila or primates. Now, some general features of transposable element. Now, first feature of transposable element is they all show movement, either directed by themselves or directed by other element. Okay. Next feature of transposable element is transposable element produces various genetic alterations upon insertion as a consequence of transposition process such as insertion, excision, duplication or translocation in the site of integration. Okay. For example, this is a DNA strand containing a transposable element. Now, when this transposable element excises itself and inserts into a new location on another strand, there will be a change in the size of this strand and also that strand where this element get inserted. In this way, transposable elements produces various genetic alterations. Also, the transposons can inactivate or alter the expression of genes by insertion within introns, exons or regulatory genes. Okay. Now, in prokaryotes such as bacteria, the transposition can occur within a chromosomal DNA or from a chromosomal DNA to a plasmid DNA. Okay? Whereas in eukaryotes, the insertion can be within the same chromosome or between the different chromosome. Okay, I repeat, in eukaryote, the transposition can be within the same chromosome or between different chromosomes. Next feature is the transposition is a non-homologous recombination. Means transfer of genes can occur between any two chromosomes, whether they are homologous or not. Okay, I repeat, the transposition is a non-homologous recombination. Means the transfer of genes can occur between any two chromosomes, whether or not they are homologous. Okay. Next feature is some transposable elements contains a gene that encodes transposase protein. This protein helps in the movement of a transposable element by excising and inserting the transposable element into another location. Okay. Now, transposable elements are classified on the basis of two criteria. First criteria is whether they contain the gene encoding the protein transposase, or second criteria is mechanism for movement. Okay. Now, on the basis of first criteria, which is whether they contain the gene encoding the protein transposase, on this basis, the transposable elements are divided into two categories: autonomous transposable elements and non-autonomous transposable elements. Autonomous transposable elements are those which can move on their own since they contain the gene encoding the protein transposase. Now, example of autonomous transposable elements are IS elements in prokaryotes and AC elements. Okay, and non-autonomous transposable elements are those which do not which require the presence of other elements in order to move, since they do not contain the gene encoding the protein transposase that is needed for transposition. So in order to move, they borrow these protein from other element. Okay, so in this way they require the other element in order to move. Now, example of non-autonomous transposable elements are DS elements. Okay, they require AC. They require the presence of AC elements in order to transpose. Next criteria is based on their mechanism for movement. On this basis, the transposable elements are classified into two categories: DNA transposons or retrotransposons. First, we'll discuss about DNA transposon. DNA transposons are those which encodes the protein transposase, and this simply move from one location to another by excising itself and inserting to another location. Okay, and this mechanism is known as cut and paste mechanism. Okay, let's understand by this diagram. In this diagram, this is a transposable element. It excises itself from this location and get itself integrated to the another location. Okay, so DNA transposons, they simply excises itself and integrates to the and inserts and integrates to the another location. Okay, they simply move by cut and paste mechanism. They cut themselves from one location and paste to another location. Okay, DNA transposons consist of a transposase gene that is flanked by the two inverted terminal repeats. This is a structure of a DNA transposons, which consists of a single transposane gene in the middle, which is flanked by two inverted terminal repeats. Okay, the transposase gene recognizes these terminal inverted repeats to perform the excision of the transposon DNA body, which is inserted into new genomic location. This transposase gene produces transposase protein, and this transposase protein recognizes terminal inverted repeat structure to excise the DNA, transposon DNA. Okay, and this type of DNA transposon is found in both prokaryote and eukaryote. Okay. Next is retrotransposon. These are those transposing elements that do not encode the protein transposon. So they produce a RNA transcript. Okay, and then this RNA transcript, with the help of reverse transcriptase enzyme, produces DNA, and this DNA gets inserted into the new location. Okay, so retrotransposons, this type of transposing elements, they do not have gene encoding protein transposase. So in this method, the DNA, it produces the RNA through transcription, and this RNA through reverse transcriptase enzyme, it produces DNA, and this DNA gets inserted into the new location. So this mechanism of transposition is known as copy paste mechanism, and this is found only in eukaryotes. Okay, that's all in this video tutorial. Thank you for joining.